Okay, let's dive in. Welcome back, everyone.
Great to be here.
Today, we're really getting into something fascinating, using AI and automation to, well, supercharge scientific research, especially when you're dealing with really complex biological data and experiments.
Right, and the basis for our discussion today is this really interesting consultation record. It focuses specifically on how you'd actually integrate AI into the day-to-day research workflow.
Almost like a blueprint.
Yeah, exactly. A kind of strategic blueprint for a future lab.
So, our mission here is to unpack that document for you. We'll look at the specific problems it flags and the solutions it puts forward, how things like structured data, AI helping out, and automation could really change lab work.
It's quite a detailed look at turning complex info into, well, actionable steps, automated ones even.
So where does this journey start, according to the record? Seems like it begins with the data itself.
That's right. The big initial challenge mentioned is just getting a handle on all the valuable information that's kind of locked up in scientific papers, mostly PDFs.
Right, like this huge library. But it's really hard for computers to, actually read and understand it properly.
Exactly. The core problem they raise is taking that massive amount of unstructured PDF content and turning it into something structured, something machines can actually work with, query efficiently, like JSON format.
And it's more than just pulling out keywords, isn't it? The document really stresses preserving the fine-grained details.
Oh, absolutely critical. It gives the example of needing to make sure experimental protocols stayed directly linked to the specific antigens they were used for.
Because if you lose that connection,
The data is basically useless for detailed analysis. You lose the context.
so how does the document suggest tackling this extraction challenge?
Well, interestingly, it doesn't propose building everything from scratch. The idea is to use mature tools that already exist. It mentions one, sort of like an advanced AI text extractor referred to as LeCheck. These tools are apparently quite good at accurately pulling out key info, the DOI, journal, author, and crucially, those specific protocols and the antigens they link to?
And getting all that into the structured JSON database you mentioned.
Precisely. That's the first step. Get the raw data structured.
But just having it structured isn't quite enough for deep science, right?
Yeah.
The document then talks about needing a layered database approach. Why the layers?
Yeah, that's a really important point it makes. Different kinds of information sort of need different homes, you could say.
Okay.
So you'd have a standard structured SQL database for the basics, author, title, journal, maybe some simple experimental parameters, stuff that's good for quick, straightforward searches.
Like, show me all papers by this author on this topic.
Exactly. Quick lookups. But then, for the really rich, complex stuff, the detailed methods descriptions, the nuances in the discussion sections, the document suggests a second layer.
That's where things like vector databases come in, or knowledge bases.
Right. Think of a vector database as being able to understand the meaning behind the words, the semantic relationships between concepts. It's not just matching exact text strings.
Ah, so it could understand that two papers describing slightly different buffering solutions for a similar experiment are actually related conceptually.
Precisely. That kind of semantic understanding is vital for the AI to really grasp the context, to do deep searches, and to match experimental setups across different papers in ways that go beyond simple keywords. It's about making the data intelligently searchable.
Okay, that makes sense. So data's clean, structured, stored smartly in these layers. What's next? Bringing in the AI for the actual analysis. The document mentions AI involves methodology preselection.
Yeah, so the idea here is you use the AI to kind of do the first big sift through all the potential methods and protocols you've now got in your structured databases.
Like an initial screening.
Exactly. Based on the research goal, the AI identifies potentially relevant candidates. It's like an AI research assistant doing a massive literature review and filtering, but based on much more complex criteria than you could do manually.
And this seems crucial from the document that the AI doesn't just decide, right? There's a calibration step.
Yes, and this is where the blend of AI and human expertise really comes in. The document is very clear. The AI's output is just a starting point, a suggestion list, if you will.
Please, refinement.
Definitely. It has to be calibrated using what the document calls custom expert knowledge data. Essentially, you're fine-tuning the AI with the lab's specific, maybe even unpublished internal know-how.
Teaching it the local tricks of the trade, basically.
Sort of, yeah. And even after that calibration, the process isn't finished. The document insists that a domain expert, A human scientist who really knows the field must review the AI's suggestion.
And check the papers it flagged.
Yes, check the relevant papers, validate the AI's reasoning, and make corrections. So the human expert isn't replaced at all. Their role shifts. It becomes more about high-level validation, guidance, and correction.
That combination AI filtering plus essential human review seems like a core theme.
It really is. It's presented as the practical way forward.
Okay, so we've gone from messy PDFs to this intelligently filtered, human-validated set of potential methods. How does that translate to the actual lab bench?
This is where the automation part really kicks in. The goal described is to automatically generate the actual step-by-step instructions for the lab instruments.
Directly from the analysis.
Directly from the structured data and the results of that AI-assisted, expert-approved analysis.
Wow. So instead of a scientist manually typing commands into a machine based on a protocol they read,
the system generates the necessary code or command sequence for the specific instrument needed for that validated protocol.
and it should be flexible too, presumably.
Yeah, the document implies it needs to be dynamic, able to generate different instruction sets for different experimental variations or needs. Adaptable.
The document also loops in some really cutting-edge tech, mentioning whole organ 3D slicing and bioprinting. How do they fit into this automated vision?
Right. That ties into getting even better data to feed back into the system. The suggestion is to potentially integrate these techniques.
So 3D slicing first.
Yeah. Whole organ 3D slicing could give you incredibly detailed, high-resolution structural maps of a tissue sample, like a really precise digital blueprint.
Okay, a very detailed map.
And then potentially bioprinting could be used afterwards, maybe for remodeling based on that blueprint or creating new tissue constructs using that detailed structural information.
And the point of combining these.
The argument in the document is that this integration provides much higher precision data about the biological system. That super detailed data then feeds back into the digital analysis and the simulation models.
Allowing for more accurate virtual experiments before you even touch a test tube.
Exactly. Better data in, better simulations, potentially fewer failed physical experiments. The document even suggests doing a pilot project just to test out integrating slicing and printing first.
It sounds like a really sophisticated feedback loop. Data analysis, my automated action, better data. But building such a complex system, there must be concerns about whether it actually works as intended. The document proposes a validation step, right? Before going fully automated.
Yes, a very practical, almost common sense step. The recommendation is to first simulate the entire workflow manually.
Manually. You mean a person does everything the automated system is supposed to do?
Pretty much. Start with the PDF, manually pull out the data, manually figure out the best protocol like the AI would, manually write down the instrument instructions, do the whole chain by hand.
And check those results carefully.
Right. And then you run the same task through the fully automated system you've built and compare the outputs side by side.
Ah, manual versus automated.
Right.
What's the goal of that comparison?
It's multifaceted. First, doing it manually might reveal best practices or crucial details you hadn't thought to automate. Second, it helps you pinpoint where the automated system might be weak or making errors. And third, it establishes a clear baseline. This is how well the process should work.
So it's about building trust and finding improvements.
Exactly. The document stresses setting up a feedback loop based on this comparison to keep refining and optimizing the automated system. You don't just flip a switch. You build, test, compare, refine.
That makes a lot of sense. So this whole picture laid out in the record takes us from unstructured data all the way to potentially automated high-precision experiments, but always with human expertise and validation woven in. Does it touch on any sort of bigger strategic points?
It does. Briefly, towards the end, it talks about the value of creating a private internal knowledge base, one that combines public literature data with the lab's own internal experimental results and know-how.
So a central brain for the whole operation.
Kind of, yeah. The intelligent repository that powers the AI analysis and decision support. And it also looks outwards, suggesting collaboration.
With other organizations.
Yes, it even gives examples like IBM, Red Hat, NVIDIA. The idea is to partner with them specifically on challenges like generating those instrument instructions automatically and optimizing the overall process.
And the goal of collaborating.
The document frames it as contributing to industry upgrade, suggesting that the methods and standards developed could potentially lift the whole field, making parts of the research pipeline faster and more standardized across different labs.
Wow. Okay, that was quite the journey through this consultation record. We really traced that path, starting with the messy PDFs.
Moving to structuring that data in those layered databases.
Then using AI for analysis.
Yeah.
But crucially, with that expert human calibration and review.
Leading to automated instrument instructions, potentially integrating advanced tech like 3D slicing and bioprinting.
And finally, that really practical validation step, comparing manual versus automated results.
It really lays out a potential roadmap and constantly emphasizes that the expert scientist isn't being replaced, but their skills are being leveraged differently, at a higher strategic level, perhaps.
And just to reiterate, this entire picture, all these steps and considerations, came directly from analyzing this single source document we've been discussing.
That's right.
So maybe the thought to leave you with, and this comes purely from the implications within this record, is this. Think about how this proposed blend, this careful mix of structured data, AI filtering, indispensable human expertise for calibration, and automated execution. How that combination, as outlined here, might fundamentally redefine the roles of data, of algorithms, and of the scientists themselves in the lab of the future. How could that shift impact not just the speed of discovery, but maybe the very kinds of questions we can effectively ask and find answers to?